Good morning, juniors, and today we are going to wrap up our study of cell metabolism with a really cool lab. So our objectives for today, explaining how photosynthesis and cellular respiration are connected and understanding that photosynthesis and respiration are chemically opposite. Quick review from the last couple of days this week. We know, and we're really good at it, photosynthesis is how plants make glucose. They make their food. They combine carbon dioxide, water, and sunlight, the sun's energy, to make oxygen and glucose. We learned on Tuesday and Wednesday a little bit about respiration, which is how all cells make ATP, the energy molecule, and how that energy molecule lets cells function. That equation looks like oxygen plus glucose combining to form carbon dioxide, water, and ATP. We talked about on Wednesday that these reactions are chemically opposite. The products of photosynthesis are the reactants for respiration. So they cycle between each other. Here's a nice little graphic showing you about the cycle of glucose and oxygen, carbon dioxide, and water. So as you can see, photosynthesis produces glucose and oxygen. They combine in the mitochondria to form ATP, that energy, and the products, carbon dioxide and water, which then are used as the reactants of photosynthesis. So all of these materials cycle through the chloroplasts and the mitochondria in plants. So today we are going to start part one of a lab because we're going to create samples that need to sit overnight. We are going to be handling a chemical called bromomethyl blue. It's an indicator that changes color based on the concentration of carbon dioxide, which has to do with the pH. So here's a picture of bromomethyl blue, the solution, and these are all the different colors that it can be based on the pH of the solution or the concentration of carbon dioxide. Just so you know, bromomethyl blue is hazardous if you drink a lot of it or ingest a lot of it. We're going to be very careful. So it's really important that you follow my instructions with lab materials the first time. There will be no warnings. You will just be assigned silent lunch. For our lab, we are going to use Alodia, which is a water plant that undergoes photosynthesis, obviously, because it's a plant, but it lives underwater. We obviously know, and we've talked about it so much, that plants always undergo respiration because after they make all that glucose with photosynthesis, they need energy, they need to do cellular respiration. We are going to prove that this is true by observing carbon dioxide production and carbon dioxide intake by Alodia, the water plant. Here's a picture of Alodia. It's floating in a sample of water. It's got some pretty thin green leaves. Your job today, we are going to do a short investigation with bromomethyl blue first before we get anywhere near the Alodia. Then we're going to set up our samples and make initial observations in the day one observations boxes in your lab handout. Those samples that we prepare today are going to have bromomethyl blue in them. That's our indicator. It's going to let us know if carbon dioxide was produced and how much when we make our observations tomorrow. So our samples are going to sit overnight and we'll check back in with them on Friday morning so we can make our observations. I just want to reiterate the fact that there are no warnings in lab, especially when we're handling chemicals. Your goggles need to be worn at all times. When you're handling bromomethyl blue, you will be wearing gloves. There are no warnings. You will just not be able to participate in our lab. You will receive a 0 and you will be assigned silent lunch. So let's have a really great lab today. If you're confused about anything in this video that I said, please go back, rewind, and make sure you understand all my instructions before we move on to the lab.